Good afternoon. Dan Guerra here from Authentic Biochemistry Studios in the Inland Pacific Northwest. I hope that everyone had a very pleasant 4th of July weekend. It was only a few days ago. And that you're ready to learn some authentic biochemistry. Now, today we're doing a video lecture. I know that you all love these because you get to see, all the images, including, of course, me delivering the lecture. But also I can go in depth because I can actually, today, for example, show you some data. It's difficult to talk about data within just audio because I really like to see the way the graphic representation works in the published papers that I'm reviewing. And today will not be an exception because we're going to do just that. So let me move this huge box out of the way. And let me move over here to where it says slideshow and do it from the beginning. All right. So again, a little bit of a comment about that picture there behind me. Let me make this thing smaller. This rock formation is right along the Snake River, almost where it joins Clearwater River in the Lewiston-Clarkston Valley. I don't know how high it is, but I'm judging from other mountains and hills that I've walked up and I've looked at over the years. It's probably about 200 feet high. And it's a beautiful uplift. I think most of it is basaltic because that's what the major rock formations are around these two river confluences. And because of its weird tilt and angle when you get close to it, it does provide a very nice shadow when you're walking. So I walk along the Snake River in the afternoon in this warmer weather. It can be pretty hot when the sun is shining. But if you go late enough in the afternoon, that big rock over there is shielding at least, maybe a quarter mile of the walk, almost a half mile maybe. So I took a picture of it and I decided that, they talk about El Capitan in California, that big, huge uplift of rock. I believe that's actually granite. But this reminds me a little bit of El Capitan. So because it's smaller by far in terms of elevation, I don't know about overall three-dimensional mass, but it's definitely much less in height. I'm going to call this wait for it, corporal, for the corporal, you see. So you have the captain at, I guess that's Yosemite, and now you have the corporal along the Snake River, where the confluence hits the Clearwater. Okay, I'm sure you wanted to know that. That picture was just taken a few days ago. Today, we're not going to talk anything more about geography or rivers or comparisons to different rock formations. We're going to talk about, as it says, amino acids and aging. And I'm doing this as a video lecture so that we can move this forward and eventually get to, yes, I know I've said it many times, and I'm going to deliver the dialectical event ontology of the aging process, grasping all the things, apprehending all the discussions we've had since way back in October of last year. It's a long haul in this arc of lectures. All right, so let's get going here. So right now we're going to be looking at a paper published in Frontiers in Pharmacology. You can see it's a publication from 2020. Let's take a look at what it says. Of course, we know a lot of this already because we've been following along, right? Aging is a natural, multifactorial process characterized by a gradual decrease in physiological functions. Those result ultimately in a reduced resistance to stress, such things as infection, such things as autoimmune responses. You also have a high vulnerability to disease in general, so the reduced resistance to stress has to do with what the immune system might do to the system. But actually the transmission and the ability for microorganisms to replicate as you age in various vascular beds or tissue beds also increases with age. So the disease can spread more quickly. And not only diseases that are infectious, also diseases like cardiovascular and, of course, cancer. You also, of course, as you age, this is inevitable, acquire an increased probability of dying. So the physical deterioration is associated with cognitive impairment in many people. And you see this in the aged population, and they call that the motoric cognitive risk, or MCR. And it's a syndrome, and it can affect up to about 10% of older people worldwide. Now, what that is an association of physical deterioration, usually in the form of sarcopenia, and then, again, some kind of cognitive decline. So, again, sarcopenia, which is an age-associated loss of skeletal muscle mass and, unfortunately, function. And that leads to a reduced physical performance. And it's independently associated with other comorbidities, including cognitive impairment, as we've been saying, where sarcopenia is recognized as a core feature of what we call frailty in the elderly, or frailty syndrome in general, which is a condition of decreased physiological reserve, mostly affecting skeletal muscle and, yes, the brain. Besides, of course, other organ systems such as the liver and the kidneys and the lungs, as you might guess. So frailty has been recently found to enhance the risk for clinically relevant dementias. Okay? That's in such dementias that occur in the background of Alzheimer's disease, being particularly associated with a decrease in physiological performance. Now, mitochondria come to play here. And they could regulate the aging process, or at least be involved in the regulation, especially in post-mitotic, highly energy-demanding tissues. And those are the ones that tend to show the most damage as we age: heart, skeletal muscle, and again, the CNS. Mitochondrial dysfunction is a crucial pathological sarcopenia. It's accumulation of mitochondrial DNA damage, that's part of it, an impaired biogenesis of mitochondria, their dynamics and quality control of gene expression primarily, and of course function related to that, as well as a reduced mitochondrial bioenergetics, there's your function, electron transport chain, oxidative phosphorylation, of course. And you get an increased oxidative stress because your mitochondria are less able to fully reduce molecular oxygen to water, thus creating reactive oxygen species. You have an age-related decrease in mitochondrial bioenergetics, coincident with the ROS, as I just said, in human brain, and also in many animal models that it's been looked at. And this is linked to deterioration of cognitive function and the onset, again, of dementia. In humans in particular, we've observed this, but also in the animal models, we'll see. So mitochondrial derangements contribute to Alzheimer's disease pathogenesis, or possibly even initiate pathological molecular cascades, which are upstream of that amyloid beta deposition that we often observe in Alzheimer's disease patients. So it's always the problem, right? What is the precondition, and then what is the consequence? In any kind of physiological process, you have to ask yourself that question. And it's not that easy to tease out one from the other, particularly when you talk about the chronic development of aging in humans. It's a long, long, long ride. And many of the earlier diseases you may pick up when you're a younger man or woman can come back to become florid and enhance the morbidities that ultimately lead to your death. This is because the immune system very, very seldom will totally lose a memory in the form of memory cells, B cells and T cells in particular, of a previous severe infection or chronic ailment, whether or not it's infection or some other kind of stress phenomena. So healthy aging, let me move this again. Research targeting mitochondria reveals that a specific mixture of essential amino acids, EAAs, enriched in branched chain amino acids, BCAAs, have been determined to potentially diminish the morbidity of aging in the animal models. Okay. So in this paper, they're going to call that BCAA-EM, enriched mixture, because you don't just have the branched chain amino acids, you also have a few other components, particularly of the TCA cycle. These are cofactors as well as some TCA cycle intermediates. Okay. So you have an activated eNOS, that's nitric oxide synthase. You also have an mTORC1, and you have PGC1 alpha. All of that is going to induce more vascularization, more aerobic environment, more protein synthesis, and more mitochondrial biogenesis, respectively, between all of those slashes in that sentence. So that's a cascade, and they study it in aged or middle-aged mice. So they want to determine if there's a preserving of cardiac and skeletal muscle, mitochondrial biogenesis, enhancing physical endurance, and increasing average lifespan. All in the mouse models, what this paper is going to be on. So BCAA-EM, that's supplementation, was able to protect mice from rosuvastatin-induced myopathy. It's a way to induce a myopathy that's a destruction of muscle fibers, and to increase oxidative fiber content, which means more mitochondria, and ameliorate, indeed, muscular dystrophy phenotype in this mutant mouse called the MDX, which is essentially a fast-aging mouse. Okay? It's a model for fast aging, and one that's particularly not only prone to, but also highly synthetically active for reactive oxygen production. Okay. So proposed molecular mechanisms from this branched chain amino acid supplementation could involve the following: de novo protein synthesis, because that mTOR pathway, reduction of protein breakdown, that's more anabolism, and notably the improvement of mitochondrial function as the PGC1 alpha and 1 beta, we'll get to that later, and an enhancement of reactive oxygen species defense reduction in the form of antioxidation of those intermediates in the one electron reduction of molecular oxygen we call ROS. Okay? So randomized controlled trial of 155 elderly malnourished patients, that was called the MATTER study, cute, huh? Show that BCAA-EM, so they used the same supplement, improved in the humans now in that particular study. General health, muscle mass, very important, and strength, as well as cognitive performance, suggesting that this branched chain amino acid supplement mediated mitochondrial bioenergetics, obviously, in the peripheral blood mononuclear cells of elderly patients. This is what we know so far, and there are other papers published on this. This paper wasn't put out that long ago, about a year ago, but there are other publications that talk about using branched chain amino acid supplementation. Okay, enhanced supplementation, that means adding in some TCA intermediates. Okay. Presumably allow the flow of carbon from amino acids into the TCA cycle using its anaplerotic activity. All right. So in this paper, that we're going to look at the data. Yay! BCAA enriched essential amino acid formulation. And here is a very specific ratio that they've determined is important. Leucine to isoleucine to valine 2 to 1 to 1. Okay. Now, if you look at the degradation pathway for those three branched chain amino acids, you can see why those ratios count. I'm not going to go through that stoichiometry. I went through it, but I didn't put it in this presentation. Sometime or other, if I'm asked in show notes or something, you can ask me to do that, I'll be glad to take you through those branched chain amino acid pathways and show you some interesting stoichiometry in terms of carbon flux and nitrogen movement. Okay, so this formulation also has TCA intermediates and cofactors, as I've been saying, And they were provided to a mouse model population with age-related mitochondrial dysfunction in the skeletal muscle and the CNS. The senescence accelerated mouse prone 8, that's got a special name. You can buy that mouse and breed it and do this kind of research yourself if you have the correct kind of laboratory and you have the right regulations for having a mouse colony, that is. And you have a lot of money, presumably either from private industry or the government, to be able to carry out this research. Senescence accelerated mouse prone 8, SAMP8 model, which progressively will develop sarcopenia, that's muscle degeneration from aging, from senescence basically. Cognitive declines, okay? Now, I put a question mark after cognitive declines because whenever I think about this, I think about my days in neuroscience labs where they talk a lot about cognition and a lot about affective behavior. And I've always questioned it. I'm skeptical about actual cognitive decline models, particularly in the rodent model. Maybe other animals you can argue for it a little bit better, maybe in some primate models, but in the rodent, I've always been very skeptical. However, they do have ways to determine this. And the way that they determine it is rather prehistoric in my way of looking at it, in terms of looking at cognition versus behavior. Yes, think of Skinner. But they still use them in neuroscience and in neuropsychiatry. So we'll go ahead and we'll take a look at this. Now, there's an AD phenotype in these SAMP8 mice that picks up between 9 and 12 months. Okay, so that includes, of course, the A beta protein, neurofibrillary tangles, and the tau protein, the whole shoot and match. Normally get from AD in human CNS at the onset of AD. Their results suggest, as maybe you'll see now, that this nutraceutical pharmacological modulation of mitochondrial metabolism, which is what they're locking into here, could correct mitochondrial dysfunction. Okay, I can argue to the point that it does look that way when you look at the data, but remember, this is a mouse model. Okay, so that is always a caveat. Okay, so here's some data. So I'm going to go ahead and stash myself way over here somewhere. Still see me, right? Yes. Hello. All right. They're showing you the mouses at ages 5, 9, 12. That's when AD kicks in this mouse. 15. You can see the mouse is starting to look a little, well, let's say it's seen better days. It looks ragged. Okay. So this is what they want to show you, that the body itself is starting to show signs of rapid aging by 15 months. Okay. In fact, it's just a progressive decline in, of course, skeletal muscle, the ability to get to oxygen saturation at high levels upon exercise. Overall, the aging phenotype that you get. There's some SAS measurements that have been done on this. The senescence-associated secretory phenotype. They've been looking at the degradation of brain cells because of neurodegeneration, skeletal muscle cells due to sarcopenia, cardiac problems, including hypertrophy, chronic kidney disease kicks in, some fatty deposits in the liver, a lot of the things you see in the elderly, as well as this cognitive aging deficit, which they also measure. So let's take a look at this then, just in graphic form. Body condition declines over time. So the body condition score, just a metric that they use for basically what the animal looks like. Its ability to gain weight, the ability for the coat to maintain a shine, that's even in there, kind of like with what happens to people's hair when they get older. And the amount of hair that's still there. You can see this mouse is losing hair, right? So it looks like it's getting older. So body condition declines with age. Body weight doesn't seem to change much, which is kind of also what we see in humans. Again, this is ad libitum feeding, so they're not becoming obese, but whereas a lot of humans might get obese for a long time into their aging process. Kyphosis is basically a bending of the spine. You know, when you get aged, the human starts to, the spine starts to deteriorate, and so the form of the person starts to bend a little bit forward. and you get that sort of curvature of the spine. And you can see that kyphosis also increases as these mice age. Okay, so hind limb clasping increases a little bit with age. They declare that has something to do with aging. Endurance, of course, declines. This has to just do with progressive reduction exercise endurance measured by a treadmill, how quickly these mice become exhausted. And then finally, the decline in working memory, and that's it's actually quantified, and it's done by a Y maze test. So they determine how quickly these mice that normally can get through a Y bifurcated maze and be able to get out of that maze. That's a cognitive skill that the mice can learn. And this, of course, related to the Alzheimer's disease, perhaps, the phenotype that they're talking about, you start to see this decline. So working memory, as expressed through a decline in working memory by the Y maze test, does decline in these mice as you move out into this 15-month age when they're looking like that. Okay. Yeah. Next slide. So Western blot, that's just showing you different months. So you're getting from younger mice to older mice. You see there's a change in the protein expression. Again, these are proteins that are run on an SDS page gel. And of course, they're separated then by molecular mass, right? Actually, by the amount of SDS that's bound to them, and that which is associated with molecular mass. And then they use antibodies for the proteins, and so that's Western blot, right? And you can see the proteins they're looking at. It's complex 5, that's the ATPase, complex 2, that's the ubiquinone, complex 4, complex 2, that was complex 3, and this is complex 2, right? The succinate dehydrogenase. There's complex 1, that's the NADH dehydrogenase, right? The first complex, where metformin seems to work. Remember that? Here's PGC1 alpha. This is a co-transcriptional activator that works with PPAR gamma. And PGC1 alpha and its friend and brother, PGC1 beta, are involved in mitochondrial biogenesis. So it's declining with age, which is relevant, right? And this is saying OPA, which we'll talk about in a minute. What else do I want to say here? So we're looking at various, so this is this data now put into graphical form. Again, the months progressing as you move on, the lighter color to the darker color here. And you can see a diminution of all of these intermitochondrial membrane complexes, okay? This is the important thing. So succinate coenzyme Q reductase, nuclear encoded mitochondrial protein dynamo related GTPase, that's what this OPA is. You can see it almost disappears as the animal ages, right? So it's a GTPase found in the intermitochondrial membrane, and it regulates mitochondrial stability, organelle stability. And because of that, energy output, because you have to have a completely integral intermitochondrial membrane complex, right? As well as the inner membrane. And the inner membrane is essential to carry out ETC and OXPHOS, and of course the proton pumping domains and whatnot. So this protein here then that we're talking about, this OPA, is then it regulates mitochondrial stability, energy output, sequesters cytochrome C. You know, if you lose cytochrome C, what happens? It's the mitochondrial associated programmed cell death of apoptosis. Right? Mutations in that gene that we're talking about here, this OPA, have been associated with optic atrophy type 1. That's why it's called OPA. And succinate dehydrogenase activities in complex 1, 4 are also shown here. Decline, decline, decline. Okay, and these are the activities. All right. So glasses, magic glasses. This is the amount of protein that's expressed in this slide here. That's just the amount of protein that's found in those bands based on densitometer readings, right? Doing a scan of this Western blot and determining how much protein is there in terms of the intensity of the signal. But then they looked at the actual enzymatic activity of complex 1, 2, the succinate dehydrogenase, that part of complex 2, Complex 4, that's the cytochrome oxidase, and then the final pathway. So the final component of the electron transport chain. And so what you see here, of course, no, actually this is citrate synthase. Sorry, there's the last one. There's the terminal oxidase, the cytochrome oxidase. And this is citrate synthase. This is part of the TCA cycle, right? And you can see that doesn't change much over time, which is interesting. And also complex 4 seems to increase, right, with age. Goes down a little bit, but increases with age. These are activities now. This is complex 2. It also goes down and comes back up a little bit. Same with complex 1, the coenzyme Q reductase. And then C1, which is the NADH reductase, you can see the same kind of thing. So you still have activity, but you have less protein. Okay? So that means the protein is no longer functional. That means the protein is functional, but the amount of it decreases. Now, why would that be? Less expression and or translocation of these proteins, either nuclear encoded genes or expression from the mitochondrial encoded sequion, okay? So that's what it's showing you. The activity fluctuates not so much of these enzymes. You still have kind of an intact DTC, even as you age. This increases in aging, increases in blue color here, but you have less protein, okay? So that's what they want you to see. It's part of the phenotype, okay? And this is in skeletal muscle. Now, here's progressive mitochondrial dysfunction impaired proteostasis in the hippocampus. Okay, we're looking at, again, the same proteins. There's complex 5, the ATPase, complex 3, 4, complex 2, complex 1. That's all the ETC. There's PGC1 alpha. You see that disappears too, right? There's that OPA for mitochondrial integrity, right? Maintaining the structure of the mitochondria, holding in cytochrome C to prevent apoptosis. This is the APP, it's the precursor protein, the amyloid precursor protein. You see that increases with age. That's part of the phenotype of the SIMP8 model, right? SIMP8 model. And what you can see here overall, okay, overall is an aging phenotype. This one other protein here is ptyrolysin metalloprotease, and it actually also declines with age, okay? GAP dehydrogenase is the way that then you can do a ratio of all of those protein amounts and divide it by GAP-DH, which does not change for the cycle of this experiment for up to 15 months. Once again, you take this data, do the densitometer readings, you do total protein, you can see that cytochrome 5 decrease, complex 5, complex 3, complex 4, that's a cytochrome oxidase. Complex 2, that's a succinate dehydrogenase. Complex 1, the NADH dehydrogenase. The OPA, that's the mitochondrial structural system protein. All decline in total protein levels, and that's significant, you see, significant as you age. But the amyloid precursor protein goes up. That's bad because that is the phenotype for Alzheimer's. And this PITRM also declines, and that's a metalloprotease, which is higher when you're younger. Okay. This is all just giving you the phenotype of the mouse. So we haven't even added yet the supplement. But what do you think we're going to do now? Now we're going to do that, right? That's the whole point. You've got to make all these measurements first, and then you look at what the supplement does. Now, I want to show you a little bit something here real quick. I'm not going to go through everything written here, but I want to show you where this PGC comes into play. So here you get transcription of SAS genes from the NF-kappa B. That's a transcription factor. And that whole process there, right, will occur if you turn on by this ATM. We talked about this enzyme. If it's phosphorylated, okay, and it's phosphorylated, this ATM protein, if you get high amounts of reactive oxygen. So when you phosphorylate ATM, you phosphorylate the protein NEMO, the IKKs. There's got to be a dimer of that. That will then activate out of kappa B, and you'll get a transcription of secretory type of pro-inflammatory cytokines. However, if that doesn't occur and you get ATM phosphorylating AKT, and then mTOR, you get a PGC1 beta dependent mitochondrial biogenesis. Okay. Now it says here senescence associated mitochondrial dysfunction could be subsequent to it, but the more you get mitochondria, the more you have nascent mitochondria, and they're fully plenum in activity, and that means there's less reactive oxygen made. So as you age, that reactive oxygen level increases. So when we're talking about this PGC1 alpha and 1 beta, and we're looking at declining with aging, you can see that that's bad because you're not going to get mitochondrial biogenesis. So that's why I pulled up this slide, and there's the reference for it. So a lot more things going on we could talk about here in terms of bioenergetics, but I'm not going to do that because I was just trying to show you where this transcription pattern functions. It's a co-adapter for transcription of PGC1 beta, and its relative PGC1 alpha. And when that declines, then you get a lot of pro-inflammatory cytokines. That's basically the senescent process. That's what I wanted to show you on the slide. Now, paper published in Frontiers of Cardiovascular Medicine, also very recently, 2020, told me the following. I wanted to look up more about PGC1 alpha. And there it is, peroxisome proliferative activated receptor gamma coactivator 1 alpha, full name, belongs to a small family of transcription coactivators, as I've said, including PGC1 beta, that's its relative, brother or sister, depending on how you want to look at it, and this other protein just called PGC1 related coactivator, that's called the PRC. Those promote via co-transcriptional chromatin remodeling mechanisms with PPAR gamma and mitochondrial physiology, enhancing all of that, okay? So PGC1 alpha was first identified as a cofactor for the nuclear hormone receptor peroxisome proliferator activator receptor gamma, just said it again, PPAR gamma, in adipocytes required for the adaptive thermogenic responses to lower temperatures. This has to do with thermogenesis related to fatty acid blocking and generating heat from the mitochondria in brown fat, particularly important in rodents, you see. Hence that foretells, because that's a blockage of the brown adipose tissue, it foretells its role in brown adipose tissue, and brown adipose tissue means it has more iron. More iron means more mitochondria. That's why it's brown in color. So PGC1 alpha is expressed in several tissue types, highly expressed in metabolically active tissues, and that includes the brown adipose tissue, and also, of course, you see it in skeletal and cardiac muscle, a lot of mitochondria. In the heart, PGC1 alpha is an essential molecule in mitochondrial biogenesis, as we've said, and muscle maturation, and it shares that role with its relative PGC1 beta. So both of them function that way, increasing mitochondria. So you just saw that aging causes that protein to decline. Okay, so that's a phenotype, declining mitochondrial number and activity. So cardiac-specific ablation of both of those co-transcription factors is, of course, embryonically lethal in mice. And that's due to, when they open up these animals, cardiomyopathy, directly linked. All right. Now, here's some data. And this is where they're adding their supplement. That's the PDOE7, right? That's the supplement that they used. You can see, first of all, here's the picture of the mice. I don't know what age exactly this is here, but an aged mice, you can see the fur is ruffled. It's got a lot of discoloring to it. The animal looks like it's aging, okay? Here's the animal on the supplement. It looks healthier, and they're claiming, from all the data I'm going to show you now, that it actually has more rejuvenation because of an increase in mitochondrial activity. So here's body condition untreated at 9 and 12 months. You can see a decline. But in the supplement-treated mice, you see that between 9 and 12 months, you don't get a decline, you get even a little bit of an increase, and there is some statistical difference there. Body weight doesn't change too much in the aging model mouse, although it does decrease significantly up to 12 months. whereas you can see it's almost steady in the treated mouse, so that they don't lose weight. And that's a good thing because, again, that last stage of losing weight is in the ultimate morbidity and decline of the animal. Right? Now, kyphosis, remember that's a spinal curvature. That didn't seem to be affected by the supplement, see? So here they showed that untreated, there was a little bit more, actually it may, because in the untreated you have an increase in kyphosis, and in the one treated with the essential amino acids, the branched chain amino acids, there's really no effect all the way up to 9 months, but then at 12 months it does seem to be a decline. So it does seem to have affected that spinal curvature. Again, if you have lower spinal curvature, right, that's good. So lower kyphosis is good, and that means this animal has a straighter spine. And they're suggesting that's also linked to this additive of branched chain amino acids. The endurance test, you can see it declines, and the wild type, or untreated, I should say, it's not a wild type because it's already a progeria mouse model, declines with age, endurance on the treadmill, whereas with the treated, it's higher at 9 months, and it goes up even a little bit at 12 months. So certainly the sustainability of endurance. Hind limb clasping goes up a little bit, which is what we had talked about back when we were looking at the controls, the phenotypic expression of what this mouse looks like, that we just went through that data. And you see it goes up a little bit, that clasping goes up a little bit between 9 and 12 months. Just the opposite, in the treated mouse, that hind limb clasping goes down. This is supposed to show a lack of an aging phenotype, okay, according to their mouse model. And finally, working memory. Again, that has to do with the Y test, right? The Y maze test. You can see that it goes down a little bit in the untreated. And here, it's much higher at 9 months because they've been treated with a supplement, presumably. And then even 3 months later, it's even higher. So this is once again trying to argue for the supplement having a positive effect on all of the different outputs that one measures phenotypically for an aging mammal, okay? Because of the supplementation of branched chain amino acids, and then those cofactors and a couple of intermediates in the TCA cycle to build up a utilization of carbon all the way through the electron transport chain and making ATP. Again, presumably this is what we're looking at, right? More data. This is an interesting. First of all, let's just read what's here. We're looking at gastrocnemius muscles of untreated and then the supplemented mice, okay? You can see the supplemented mice. This muscle looks more filled out. It's not, there's no damage. This one has partial damage on this side, so it's like probably some kind of sarcopenia going on here. Whereas this little fragment here, it doesn't look like there's nearly as much sarcopenia on that rough edge. And this is muscle weight. You can see that the treated has a higher weight. Likewise, this is a staining. They're looking at the cytochrome oxidase 4, right? That's the terminal oxidase, the electron transport chain. You can see the untreated, it's kind of spotty in staining. And the treated, this looks like really healthy muscle, meaning a lot of active mitochondria. And here you're looking at the the S6 kinase, okay? So this protein is part of the anabolic pathway, and the phosphorylated form of it, okay, is considered to be the one that's necessary to induce anabolism in mammals. And you can see indeed that it's increased upon the treatment. You have higher levels of phosphorylated S6 kinase, okay? Here are all the same proteins we looked at before. These are Western blots. You can look at untreated. Remember the decline in all of these complex electron transport chain complex proteins. Decline, decline, decline. There's your standard GAP dehydrogenase. That's just what you do the ratios for the two different protein concentrations. But look at here. With all the supplements, you get a robust increase in total protein on the Western blot. Right? Almost all of these look better with treatment, right? And here's this data then put into histogram. You can see every one of these red bars, every one of these electron transport chain protein levels is better when you gave them the supplement with the branched chain amino acids, the co-substrates and the TCA intermediates. That's that total supplement package, right? And it's the same thing we're looking at here. This is the NRF, and that's also a really important thing. That particular protein is a transcription factor that you need in muscle tissue, and you can see that it also goes up when you give it the supplement as compared to the non-supplemented control. All positive, right? Then we're looking at the enzymatic activities here, and here you do see a good promotion of activity, right? This red bar is all of those complexes, and that's what you're looking at here, enzymatic activity. They're all better with the supplement. Here's a Western blot showing you treated versus untreated, and once again, you're looking at an increase in, this is particularly complex 3, right? And so this is really important. You're also looking at complex 3 forming a super complex, which is what it does in fully functional electron transport chain. You make a super complex, that is, you make a polyprotein of complex 3. And you can see, and this is part of showing mitochondrial integrity and full fidelity of the mitochondria. And you see here, I love it, you see an increase in that complex with the addition of the branched amino acid supplementation. Okay, so this is looking at all the markers, looking at the muscle tissue itself, looking at the electron transport chain, the protein level and the enzymatic level, that putting in this additive is in looking at the mice themselves, their overall and their endurance. It looks like this is enhancing their longevity, right? With all the parameters, even the cognitive parameter. That's what they're saying here. Okay. Now this indeed, that was in muscle. Now we're going to finally look at the hippocampus. Remember, this is going to be the CNS story, right? We're looking at the CNS. Here's treated versus untreated, the same kind of thing. The kinase that's involved in anabolic pathways, right? You can see here that you get more phosphorylation in the treated mouse with the branched chain amino acids, and that's shown here. And likewise, you're looking at the same proteins looked at before. Also some interesting ones like SIRT1, which of course is a deacetylase. That goes up in the treated. That's a story I'd like to follow up on, actually. Sorry about that. And because sirtuins, you see, sirtuins are deacetylases, and that would tend to make you, if you have a sirtuin activity, it deacetylates chromatin, meaning you have less overall gene expression, and that seems to go up with the treatment. So that's kind of curious. But the OPA goes up, which means more mitochondrial integrity. That's a good thing. PGC1 alpha goes up, right, in the hippocampus. That's also a good thing with this treatment. Means more functional mitochondria, more functional CNS, and then all these complex of electron transport chain. And there's all their activities or protein levels, and here's some of their, I guess it's also protein. My glasses back on. Yeah, this is just looking at some of those other proteins that we were just discussing. So you see, overall, this experiment has shown that if you add branched chain amino acids and a few supplements that drive the tricarboxylic acid cycle, that you will enhance mitochondrial respiration as shown by an increase in the functionality and activity of all the electron transport chain proteins in the inner mitochondrial membrane, as well as more mitochondria being more active because of this carbon source, okay, using branched chain amino acids. Now, this again comes from literature that's been in the peer-reviewed experimental papers for at least 20 years, looking at animal models. For some reason, giving branched chain amino acids, which are essential amino acids, the ones they're using there, so we don't synthesize those, supplying those at higher levels has been touted as being good for decreasing some of the morbidities in aging in humans. Papers like this would tend to reify that nutraceutical sort of argument, right? Now, again, the caveats here is this is a mouse model paper. They already have a mouse model that they're using, which goes into advanced aging and gets an AD phenotype as it ages. So this is different from a wild type mouse. So already you've got a model system. Secondarily, remember you're giving these animals sufficient amounts of these branched chain amino acids that would have to be replicated in a human at such high levels. And the same metabolic fate would have to also be incurred in humans versus this mouse model system. So that's a lot of, what do I want to say, speculation on the part of research like this. We would have to show that you indeed could increase the overall activity of the mitochondria and make them more functional by adding these branched chain amino acids in a human study. Now, you did see that one study where people have done this kind of work. It did look like it was increasing the healthy phenotype of elderly adults, but that was a very small N number for that study, but still it did appear that way. Well, this mouse model here really gave us a lot of, I would say, pretty definitive data of what's occurring at the biochemical phenotypical level. Right? All right. So I just want to show you briefly here the pathways we like to talk about in amino acid metabolism. So when you're taking glycine or serine and you're doing the catabolic pathway, you will enter the core glycolytic pathway at 3-phosphoglyceric acid level. And of course, at 3-PGA, that could be used for gluconeogenesis or indeed for glycolysis. Right? So this is where the carbon ends up in the glycolytic pathway for glycine and serine. Same thing here, glycine and serine can also be used to make pyruvate, as can alanine. We talked a lot about that in previous lectures, that all you need is the transaminase reactions, and because the carbon skeleton for pyruvate is alanine. Okay, so look at the other amino acids. They can add their carbon into glutamic acid, proline, glutamine, of course, by deamination, arginine, and histidine will all, upon catabolic pathways, make glutamic acid, which, of course, because of transamination, will make alpha-ketoglutarate enter the TCA cycle. Likewise, at the oxaloacetic acid level, asparagine converted to aspartic acid, you'll enter carbon there, and that can ultimately be gluconeogenic above the pyruvate level, right, at the pepcarboxykinase level. All right, so threonine can be converted glycine to serine. That is always the same there. That's that glycine pathway that I've shown you many times. It's actually linked to folic acid metabolism, making 3-PGA. So the first one is non-essential amino acids. Here are the essential. Threonine is essential. Here are several more. There's lysine, tyrosine, isoleucine, leucine, and tryptophan. They can all form acetoacetate, which is a ketone body. That can, of course, be converted to acetyl-CoA after addition of coenzyme A. Cysteine can be converted to pyruvate, shown here. Sorry. Tryptophan can be converted to alanine, and of course, to pyruvate. Threonine to glycine and serine, already talked about. Threonine also can be directly converted to pyruvic acid. The phenylpropanes, phenylalanine, and tyrosine can be directly converted to fumarate. Threonine and methionine to alpha-KG. And valine and isoleucine, those two very important branched chain amino acids we were just looking in that study, can be converted to propionyl-CoA, and then that can be converted to succinyl-CoA, and then a TCA cycle. Right? This is all bioenergetics, showing you what happens at the fate of these amino acids. Really important for you to understand whether or not this is coherent with the paper we just looked at. So, to remind everybody, the essential amino acids are the following: histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Notice tyrosine isn't in here because you can actually make tyrosine from phenylalanine. Okay? And tyrosine, of course, is a really critical amino acid for neurotransmitter production, for example. Non-essential amino acids, they're the ones that are left over, and these are ones that you synthesize, of course. Okay. Arginine, cysteine, and tyrosine are considered to be conditional essential. And again, they can become rate limiting. If they become rate limiting, if there's very low levels, they can be reconverted because of transamination reactions. And arginine and tyrosine are also involved in a couple of other important pathways, arginine in the urea cycle, and of course in nitric oxide metabolism. And tyrosine is linked up into C1 metabolism. Okay, but those are called conditionally essential. So this is just showing you, whoops, sorry, let's go back. That would be better. The transamination reaction, also known as the aminotransferase reaction. This will take any given amino acid. Okay, so start here with R1 with an R group here. This is an amino acid. And when you take an alpha keto acid and react it with an amino acid, depending on the enzyme catalyst itself, because they're specific for specific amino acids, these transaminases are also known as aminotransferases. Then you make a new carboxylic acid, okay, from the amino acid that just passed its amino group onto this substrate carboxylic acid, and you form now a new amino acid. Okay, so this is what transamination reactions do. They're freely reversible, depending just on substrate and product concentrations and utilization of those amino acids and carboxylic acids in pathways. What kind of pathways? Protein synthesis for the amino acids, for example, or for degradation in bioenergetics, and of course the alpha keto acids for a plenum of biochemical pathways. Here's one showing you just one specific. Alanine alpha ketoglutarate will make, of course, pyruvic acid and glutamate. And this is a very common transaminase reaction associated with the mitochondria. All right, I'm going to stop here because this is going to start showing you some of the real biochemistry of amino acid catabolism. By real, I mean we're going to do some arrow pushing. We're going to show you reaction mechanisms, kind of thing I like to do in a graduate biochemistry course. And of course, authentic biochemistry is just that, plus. So I will continue this, and I will go through the amino acid DEG pathways, because I want you to ultimately see where all those branched chain amino acids function physiologically and biochemically, and how that may relate more directly to what we saw with that mouse study I just went through. Right? We just went through that mouse study, and then what I like to do is show you how that fits into how that fits into what we know about amino acid catabolism. End show. All right, whatever that is, I don't know. I'm going to get rid of that. All right, so I think that we've done our duty for today. We've discussed a paper that seems to suggest that mitochondria, the longer that they are maintained as active and functional in senescing skeletal muscle, cardiac muscle, and indeed the central nervous system, the longer it seems that, at least in the mouse model, we can prolong a non-morbid, healthy aging phenotype. Okay, now remember the caveat, that was a mouse model, and it's already a mutant mouse that ages relatively rapidly and gives us the AD phenotype at the end. That's why it, in fact, was bred. Okay, so it's not a wild-type mouse. That's one big skeptical caveat, comparing that to what can happen with just regular humans aging. And of course, the human metabolism, even though those amino acids are also essential in humans, is going to be unique because humans have a unique processing ability. So the modality of biochemical pathways are different within, actually across species and even within individuals because of epigenetic considerations, as we've talked a lot about in the past. So I think I'm ready to sign off for now. And when I do that, I will go ahead and post this on our YouTube channel. So I hope that you got good information from this presentation. It was fun doing it. And I like to do these primary research papers, and that was a new one. We'll do more of those as we drive down into the dialectical finale of the event ontology, which is human aging. And I'm going to do that because I want to see some primary papers that will throw a little bit of ambiguity into what will be my final considerations and points that I want to drive home by describing those ultimate morbid conditions that lead to tissue degeneration, mutation, epigenetic modification that lead from morbidity to mortality, thus the final stages of the life pattern in humans. So this is Dr. Dan Guerra, very much alive on the 7th of July, 2021. Wishing you all a good day, and bye for now.